In this chapter, we're going to begin our discussion on viruses. So to begin with, let's go back and do a little bit of remembering of the history. The term virus first originates from the word in Latin that means poison. And you may recall that Louis Pasteur is the one credited with first using the term virus, and he did so to describe that causative agent when he was doing his research for rabies. Again, the first virus that was actually discovered was the one that was associated with the tobacco mosaic disease. Now in 1890s, they just realized again that there was a causative agent for the disease, and then in about 1930s, they actually isolated that causative agent and realized what a virus was. Now, why did it take a span of 40 years or so? Because unlike bacteria and some of the eukaryotic microbes, in which a microscope is able to help us identify, look for, in that kind of scenario, the viruses are so small that that standard microscope didn't work. So it took the discovery of the electron microscope in order for them to actually visualize and see and isolate that first virus. So from the 1930s up, we had a steady flow of information and research on viruses, and by the time 1950s rolled around, the field of virology itself had been established. So let's talk a little bit about viruses because they're such a unique pathogen in and of themselves. One of the first things that you really need to understand about a virus is that they are simple acellular particles. Now, by the biological science's definition of life beginning at the cellular level, there's a huge discussion going on if viruses are living or not. Technically, they're not at the cellular level, they're acellular. They cannot do some features what it would mean to be a living thing. So right now, viruses are considered to be non-living particles. Now, a couple of things to know. They're extremely small. They're typically going to be less than 500 nanometers, which means that's 10 times less than the size of a bacteria and smaller. Viruses are going to be able to infect both eukaryotic cells and bacterial cells. When they infect a bacterial cell, we actually refer to them as bacteriophages. Viruses multiply. We don't actually say replicate. Why? Because they are completely dependent upon a host cell in order to multiply or make more of themselves. So they are obligate intracellular parasites. Viruses and those bacteriophages that we're going to talk about, they're not capable of any kind of metabolic activity. Why? Because they lack that machinery. They don't have any of those organisms, those organelles such as ribosomes needed for making proteins, or to be able to replicate DNA or replicate RNA. So they're going to have to get inside a host cell, and they're going to have to literally take over the host cell's genetic mechanisms to be able to synthesize and assemble new viral particles. And again, for 6 and 7, they don't have that machinery, so they're not going to be able to make proteins. They're not going to be able to have the enzymes and stuff necessary for metabolic processes. But it should be noted that viruses are going to be able to exist extracellularly, so outside of the cell, or intracellular. In that extracellular state, so when they're outside of the cell, the virus is actually called a virion because it's not able to undergo that multiplication process. So let's look a little bit here at the viral structure. So the virus is going to consist of genetic material. That's going to either be that nucleic acid, it's going to be RNA or DNA. Around that genetic material, there's going to be a protein coat called a capsid. And collectively, that protein coat in genetic materials is known as the nucleocapsid. And then some viruses are going to have an envelope, a third structure, if you will, that is surrounding the entire nucleocapsid. So let's take a little bit deeper dive into each of these components. So let's start first with that capsid or that protein coat. That protein coat is going to be made up of many individual proteins known as capsomeres. And those capsomeres are going to completely surround the genetic material of DNA or RNA. And that capsid or protein coat is going to come into one of 3 basic shapes, which we call a helical, polyhedral, and complex. And that helps us to identify a virus because we often identify them based on that capsid shape. So 3 different capsid shapes as stated. So the first one is the helical. This one, those capsimers, those individual proteins are going to form a ribbon-like shape, spiral shape. Think, if you will, of a spring, okay, like a spring that you find in a pen or anything else like that is going to be the helical shape of that capsid, and then the genetic material is going to be down in the center of it. That spring, as you may recall, that helical shape, like many springs, some of them are going to be flexible and some of them are going to be very rigid. Same thing can happen with the helical capsid. Now the polyhedral is a geometric shape for a polyhedron, and that's going to have 20 facets or sides if it in 12 corners. So this is like almost a diamond shape, if you can think of that. And so polyhedrals, that's going to be like a herpes virus and polio virus. Okay, they belong to this category. And then the third category is what we call a complex virus, and we call it complex because we don't like to use the term miscellaneous in science, and the complex virus just means that it has unusual shapes. And you're going to see some unusual shapes, especially when we talk about the bacteriophage, which is that virus that gets inside bacteria. And again, this just means that there's no central look to these. Okay, they're all going to be a little bit random and unusual in their shape. So those are the capsid. Second component that's found in all viruses is the genetic material. Now, unlike the eukaryotic cell, like your cell, which has DNA and RNA, viruses will not do that. They're only going to have one type of genetic material, DNA or RNA, but they will not have both. The other thing that is unique is that the DNA and RNA, it could either be double-stranded or single-stranded. So DNA can be double-stranded or single-stranded. RNA can be double-stranded or single-stranded. So it's only going to have one form of that genetic material. So the parvovirus is going to be a single-stranded DNA. Double-stranded DNAs, herpes virus, adenoviruses, pox viruses, things along those lines. Single-stranded RNA, because it's only going to be once in like your right and left hand, we call them plus and minuses. So you can have retroviruses, rhabdoviruses, picornaviruses, and then there's going to be double-stranded RNA, which is a reovirus. Now, what you may recognize real quick here is that the double-stranded DNA, there's a lot of examples of those causing diseases, and single-stranded, there's a lot of those that cause diseases. Why? Because think about it. Humans have double-stranded DNA and single-stranded RNA. So if the virus has genetic material that's closely resembling the human genetic material, then it's going to be easier for it to take over the cell, get inside, obviously, and cause damage. The third component that we talked about was the envelope. Again, want to stress this is only found in some viruses. And so what it is when the virus gets into a host cell, the virus, in order to cause damage and to infect others, is going to have to get out of that host cell after it has multiplied and made more of itself. So it's got a couple of different ways. Sometimes it just bursts and the cell releases everything, but in other instances it does what we call budding processes, and it pushes its way through the cell's membrane. And you might recall that that cell membrane in a host cell is going to be a phospholipid bilayer. So when it pushes through that phospholipid bilayer, it actually coats its capsid with that phospholipid bilayer, and then that forms this envelope. And then on some of those envelopes, you're going to have different proteins sticking out, and those proteins, we refer to them as spikes. Now, because of the fact that that envelope originates from its host cell, it will serve as an advantage for enveloped viruses because they're going to look very similar to its host cells, so the body's not going to recognize it as quick. Okay? All right, so those are our three pieces of the viral structure. Now, viral multiplication is a in-depth kind of complex process. Why? Because again, they're obligate parasites, and so they have to rely on their host cell in order for them to be able to make those proteins and assemble it. So they're intracellular obligate parasites. They're going to invade eukaryotic cells, and that's going to be very different from when they invade prokaryotic or bacterial cells. So you will need to know the steps of multiplication in both the animal and in the bacterial cells, and that's a separate video, so look for that one as well. One last thing on viruses themselves is there is a term provirus, and this is referring to a virus that has the ability to take a piece of its genetic material and incorporate it into the DNA of its host cell. And then that viral genetic piece will be incorporated in that DNA, and it will stay there. And it may not be active. It may be that the host never even recognizes that it has an infection until something occurs to trigger it. And then it will go from being in that dormant or latent state into an active state. And then at that time, it will replicate, multiply, and then it will lead to the whatever it may be doing to that host if it's a disease-causing virus. This is often associated with retroviruses. There are two other items at the end of the chapter that I just want to point out, terms. These are not viruses, but they're put here because they are also non-cellular, very small agents that can cause harm and disease in humans. One is what we call viroids. Please don't get that term mixed up with virion. Remember, virion is the term for the virus that is outside of a cell. This is a viroid. And in the case of viroids, it's fragments of RNA, okay, that can then cause and get into, and generally it's the plants that are going to have problems with this. They get into that replication process, and then they can cause plants to become diseased and die. The other one is more in the human, and this one is what we call prions, and prions are not part of genetic material. So remember, viroids are going to be the RNA, and then the provirus, the one we just talked, which was a latent virus, it was a virus, was DNA, but prions are actually what they call proteinaceous infectious particles, and they are thought to be just small pieces, and we say of proteins, but in actuality there are only so many numbers of amino acids, very, very small numbers of amino acids, that they probably aren't even a true protein, but they can get into humans and cause things, and in animals, and cause things such as mad cow disease. And in humans, it's going to be neurological diseases. Now, how that occurs is because an infected animal, this gets into their neurological tissue, so the brain and the spinal tissue, and then sometimes in poor meat processing, then that brain or spinal tissue can get into the meat that is processed like ground beef for human consumptions, and then humans eat it, and then it will cause these neurological diseases. So that is just the introduction to the viruses. The multiplication cycle of animal viruses and bacteriophages is in another video. Please look for it as well.